So, hello. I will make the presentation in English. Sorry for this. My name is Amalia, and I'm new. Here. Okay, sorry. Okay. So, yes, my name is Amalia, and I'm new PhD student in the lab of José Manuel González Navajas. And today I will present you some results of my PhD thesis that is about the salt-Th17 axis in the liver inflammation and cancer. So, hepatocellular carcinoma is a classical inflammation cancer. Approximately 90% of the cases are associated with prolonged inflammation caused by chronic liver damage, including cell stress and cell death that leads to compensatory proliferation, following by fibrosis and cirrhosis. The major risk factors include viral infection, metabolic disease like fatty liver disease, and extensive alcohol intake. As we can understand, the immune system plays an important role in all these procedures, and both the adaptive and the innate immune system are important for the detection and elimination of transport cells. Although in the setting of chronic liver damage, this immune network is also deregulated, and this is why the surveillance of apparent cells is also not working well, and all these events collectively leads to the tumor initiation and hepatocellular carcinoma. In this concept of chronic inflammation, an immune cell type that has been found to have an active role are the Th17 cells. The Th17 cells is a subset of pro-inflammatory CD4 T helper cells. The CD4 T cells, when they get activated, they can be differentiated into Th17 cells by the stimulation of IL-6, TGF-beta, and IL-23 that there are in the microenvironment, and they are characterized mainly by the production of IL-17, but also IL-22. They have an important role against the defense against extracellular bacteria and fungi, but they have been present also in the tumor microenvironment or several tumors. And there they can have tumor-promoting properties, mainly by inducing angiogenesis and by recruiting pro-tumoral cells like neutrophils, macrophages, and myeloid suppressor cells, but they can have also in some other tumors tumor protective properties, like by producing interferon gamma and by recruiting cytotoxic cells. And now, specifically in the context of hepatocellular carcinoma, the Th17 cells have been found enriched in the tumor of humans and are also correlated with poor survival and poor prognosis. The specific tumor promoting mechanism is not yet well understood and probably is not neither Catholic, but have been found that they can act direct to the tumor cells by, for example, inducing the IL-6, JAK2, STAT3 pathways that leads to the survival of tumor cells, or indirectly by inducing suppressive immune checkpoint molecules in the macrophages that later on can suppress the T cells. Also has been associated with the IL-17 has been found to be important in alcohol-induced hepatocellular carcinoma, where the signaling, where they found important role of IL-17 signaling in steatotic hepatocytes, and also has been found to have an important role in the process from non-alcoholic fatty liver disease to non-alcoholic steatohepatitis and hepatocellular carcinoma. Now, some years ago, two independent research groups found yes, that the salt, the modest increase of salt concentrations can enhance the differentiation of Th17 cells in vitro in humans and mouse cells. And the high salt diet can induce the development of pathogenic Th17 cells and accelerate autoimmune disease in vivo. From a molecular point of view, this is quite well understood and includes the MAP kinase pathways, MAP kinase and NFAT pathways that leads to the activation of the signal of SGK1 that at the end induce the expression of Rho gamma T, which is the crucial transcription factor of Th17 cells. It's important for the differentiation and the stability of the cells. So based on what has been said before, we wanted to study the role of the salt Th17 axis in the liver disease. So we hypothesized that extensive salt intake that may lead to the accumulation of pathogenic Th17 cells can increase the severity of liver disease, chronic liver disease, and may facilitate like this the tumor development or also growth. So to test our hypothesis, we designed some specific objectives. First, we would like to see whether high salt diet can accelerate or increase the severity of HCC, hepatocellular carcinoma, in mice. And secondly, we would like to see whether low salt dietary can improve the immunological profile of patients with compensated cirrhosis. So for our first objective, we used the DEN-Induced acute liver injury model, the Diethylnitrosamine. It's a carcinogenic that is used widely to induce hepatocellular carcinoma, and this because simulates the process of chronic injury, necroinflammation, regeneration, and malignancy that has been seen in humans. So specifically, what we did, we used male mice, and we divide them into groups receiving either normal or high salt diet. And after 3 weeks, these mice were injected with DEN once. And after 2 days, they were sacrificed. From the sacrifice, we isolate the liver and the tumor. In the liver, we perform histological analysis and mRNA analysis, And in the blood, we measure some serum parameters, some hematological parameters. Okay. So from our results, we found that the high salt diet induced the liver damage that is induced by increase the liver damage that is induced by the DEN, as evidence of the ALT, where in the group of high salt diet is increased in comparison with normal diet. And also, we found similar results in the same line by histological analysis, where in the high salt diets in plus DEN group, we saw bigger percentage of necrotic areas, as you can see in these images, in comparison again with a normal diet, but also with a high salt diet without them. Okay. As regarding Th17 cells, we found by histological analysis that in the high salt diet group that were injected with them, there are more Th17 cells in the liver. We found, yes, more presence of Th17 cells in the liver. That was also statistical significant in comparison with the normal diet and the high salt diet without them. In the same line with this, we perform mRNA analysis, and we find that the the raw gamma T, that, as I said before, it's the crucial transcription factor for Th17 cells, is the only one that is induced in the high salt diet, while the rest of the transcription factors that are crucial for the rest of the T helper subset are all of them decrease. This means that this more specific inflammation that is used by Th17 cells and not by other cells. Okay. Then we want to check the involvement of Th17 cells also in the process of hepatocellular carcinoma. And for this, we use again the same model. We did the same. We divide the mice in normal and high salt diet food. After 2 weeks, we perform a DEN injection. And after 1 week, were followed. We perform 10 weekly CCL4 injections. The CCL4 in this case is used to induce fibrosis and tumor growth. And the mice were sacrificed at weeks 36. And in this analysis, we isolate the blood and the liver again. In the blood, we measure the number and the size of the tumors, and we keep a piece for histology, Western blot, RNA analysis, and the rest of the cells we used for non-parenchymal single-cell isolation, and they were stimulated in vitro, and then we perform FACS analysis. And in the blood, again, we perform FACS analysis, and we measure some hepatological parameters. And what we found from this analysis, it was first that in the DEN group, all the mice develop tumors, both in normal diet and high salt diet. Surprisingly, also 2 mice in the high salt diet without DEN develop tumors. And regarding the tumor growth, we saw that the group of high salt diet had more tumors in comparison with a normal diet, but also bigger tumors. Still, these results are not statistically important, and for this, we would like to increase the number of the mice of this experiment, so we improve the statistics. We hope so. We can improve the statistics. Regarding the Th17 cells that we measure by flow cytometry, we see that in the high salt diet, there are more Th17 cells. The percentage of Th17 are more. Again, here the statistic is not significantly important, but there is a trend, an important trend. And okay. While interferon gamma, that is the Th1-related cytokine, is not affected. And again, here, I think this is nice because it means that it's an inflammation that is induced by Th17 cells and not from any other immune cells that probably could have been also induced because of the injury, of the liver injury and the hepatocellular carcinoma. So it's more specific. Okay, regarding the blood analysis, we didn't see so much difference between in the parameters that that they show liver damage, like ALT and ALT and ALP. Although again there is a trend of increasement in the high salt diet. Although we saw a difference in the total protein and in globulin, which are in both cases induced. And while the ratio of albumin globulin is reduced, which is evidence of probably systemic inflammation, that there is inflammation, induced inflammation in the high salt diet group. And the cholesterol is not really affected. Now, yes, this is more or less the data, the important data that we have from the mouse model. And now I will move to the second objective, which was whether reducing dietary salt intake can improve the immunological profile of patients with compensated cirrhosis. So to do this, we perform a non-drug quasi experimental study in patients with compensated cirrhosis. The patients in their first visit, they had to provide us with dietary diet history questionnaire. Also, they were interviewed with nutritionists. And at that day, blood, serum, and 24 hours urine sample was collected. After the visit, the nutritionist was calculating the daily salt intakes from their foods, from the food's habits, yes. And we share the groups. We divide the patients in 3 groups. In the ones that they have less than 5 grams daily intake, which is the recommended by the health authorities, by the World Health Organization values for salt. The ones that they have between 5 and 9, which we consider them high salt intake, and the ones that they have more than 9, which is the very high concentration. Then the nutritionist, in accord with the hepatologist of the team, was providing personalized diets and guidelines to reduce salt intake in these 2 groups, in the ones with high salt intake, daily intake. And the patients had also to record twice per week, randomly, their snacks and meals for 3 months. And after 3 months, they were coming back, blood and 24-hour urine sample was collected again. They were providing they were giving their questionnaires in the nutritionist, and she was calculating whether they follow or not the recommendation for low salt intake. Regarding the biological samples, yes, the blood, we isolate CD4 T cells by magnetic beads. We keep a part for mRNA analysis, and the rest of the cells, we stimulate them in vitro. A part of them was used for flow cytometry analysis, and the rest of the cells for 24-hour stimulation, and then for cytokine analysis in the supernatant by ELISA. So from our results, we found that, yes, that there is a positive correlation between salt intake and Th17 cells in the blood. Yes, also the ratio of Th17 to Th1 had also an even stronger positive correlation with the salt intake, while the interferon gamma positive cells have a negative correlation. These data were also confirmed by when we repeat the analysis with the natrium of the of the concentration of sodium, with the concentration of sodium in the 24 hours urine sample. And this is nice because it seems that the estimated values of salt intake by the food are relevant, and yes, are relevant, that the patients don't lie. Okay. Then we perform the same type of analysis in the cytokine production by the CD4 T cells. And similarly, the Th17-related cytokines, the IL-17, the colon stimulator factor, the IL-6, and yes, the TNF, they present, again, a positive correlation between salt intake and their concentration, while the Th1 and Treg cytokines, they don't change, they are not affected by salt. Then by using the same data, we perform a different kind of analysis. We use the medium volume of, the medium value of salt intake, and we share the patients in 2 groups. The ones that they consume low, or they have low, no consume, they have low salt intake, and in the ones that they have high salt intakes. And again, we see here that in the group of high salt intake, there are more Th17 cells in comparison with low. Similarly, the ratio Th17-Th1 cells is increased statistically in the high salt intake group, while the interferon gamma positive cells, the Th1 cells, are again kind of reduced in the high salt intake group, but it's not so strong as the IL-17, Th17 cells. Okay. No. Okay. Now, importantly, from the analysis that we did after the diet, so in the second visit, firstly, we saw that the patients reduced significantly the values of daily salt intake. This means that they follow the recommendations. And more importantly, when we compare individually every patient in the visit 1 to the visit 2, this leads to significant reduction in the numbers of circulating Th17 cells. So when they reduce their salt, they also reduce the circulating Th17 cells. Okay, so this is more or less what we have. So in conclusion, we can say that high sodium intake aggravates the liver damage and enhances Th17 associated response during acute liver injury induced by DEN, and this is important because it's the first step for HCC initiation. High salt diet seems to aggravate hepatocellular carcinoma, tumor growth, and correlates probably with systemic inflammation in mice. And salt intake in the humans upregulate the Th17 depending inflammatory profile of patients with compensated cirrhosis. In the future, we would like to test our hypothesis in a non-alcoholic liver disease, fatty liver disease model that leads to NASH and hepatocellular carcinoma. We would like also to investigate whether by modifying the immunological network, salt intake can also affect the response to immunotherapy, like immune checkpoint inhibitors. And also we would like to perform some in vitro experiments to investigate the mechanism of sodium impacting the hepatic tumor microenvironment. And thank you. Thank you for your attention. Yes, hello.
Congratulations on my presentation.
Thank you.
I have a couple of questions. First, because I didn't know, is there any immunological relationship between salt intake and salt intake and worse evolution of liver disease or cellular carcinoma?
In the compensated patients of cirrhosis, not really. The only thing that is found is that from patients with hepatocellular carcinoma, that they consume, that they have higher salt intake, but there is not really correlations there, yes. But in the decompensated, yes, and this is why we didn't include also in the study, because they are they don't have to, they are salt restricted. But in the compensated, no.
They develop acidity.
Yes, in the ones of, yes.
They are also going to work. They didn't.
You mean, well, in general.
Are going worse if they have high intake of salt?
No, this is not proved. No.
My second question is, this relationship between salt intake and Th17 response in that looks potential in hepatocellular carcinoma has been also demonstrated in other tumors? Involved in the immunological response to different.
Yeah, not really. Specifically, not really in inflammation-related cancers. There are two papers that they published recently, but in fact they use xenografts. So I wouldn't say that this is a real evidence yet. So yes, there are two. In one was, but are xenografts. I think the one was, I don't remember. But yes, only this. But yeah, not in any. Yes, in inflammation. Yeah, I don't remember what. I don't remember. Yes.
Thanks very much for your nice presentation.
Thank you.
Now, you're always talking about intake, salt intake. But really, what does it matter? Salt intake or serum sodium level?
Serum blood levels. Blood levels are always stable, so I don't think. For me, probably, if there is accumulation in the liver, for example, that in the mice you can see this accumulation of sodium in the liver.
Yes, but when we are talking about patients,
Yeah.
particularly cirrhotic patients, they their sodium levels fluctuate, and sometimes they have low serum sodium levels.
Okay.
They have expression of sodium which varies depending on their state. So really, is the intake or the serum sodium level what matters?
Okay. Yeah, I guess both. I guess.
It should be good.
Yeah. Okay, I understand. Okay. Yes. Yes. We have to. Yeah, we have to think about. We have to think about this. Yes. Yeah.
I might be wrong, but even in this compensated patient, the blood sodium level doesn't change much. It's very stable because the kidney is functioning according to water and is expression of sodium. So I think more than in blood, in urine, sodium in urine, maybe better marker.
So again, the development of hepatocellular carcinoma is not always taking place in compensated patients.
No, sometimes in compensated, I mean, but from compensated patient, you can also get that hepatocellular carcinoma.
Yes. Yes.
So that's a good question. How sodium in vivo is using this administer? Is circulating in cells, is in the gut? Maybe the important is the salt concentration in the gut. And then you know that an approach is the differentiation of tissues, and then they go and can go to the liver to the postal situation. There's also a question where is this in vivo using the differentiation of the tissue, the gut, the liver, the blood.
Yeah, this is true.
We don't know.
And again, patients with advanced cirrhosis, they usually have less immunological response than other patients. In fact, they need less immunosuppression when they are transplanted because they have a low immunosuppression level. So is there any correlation between the immunosuppression state of these patients and the development of the hepatocellular carcinoma?
Yes. Normally it's unconscious.
And again, finally, one of the goals of patients
Sorry, I didn't understand the second part. I didn't listen. I didn't listen the second part. I didn't understand. Sorry. Yeah, probably if we induce pathogenic Th17 driven inflammation, of course, this probably can make the situation worse than for them. Can probably make it faster, like the initial.
Most of them do not have cirrhosis.
Yeah, but I think, again, I mean, our study, in the patient study is in cirrhosis, but in the context of HCC, that inflammation is important, I think yes, that is important. That can be correlated, yeah. Ye- Yes, exactly, that's why, yeah. Okay, thank you.